I think the important potential implications of this work is that not all amyloid is the same. So there are variations, and we see variations from patient to patient. And this may have significance for explaining a bunch of observations. For example, the main observation is that there's not a very strong correlation between the total deposition of amyloid in brain tissue and the extent of cognitive impairment or the rate of progression to Alzheimer's disease. So consequently, just measuring the total amyloid load has limited predictive value. So there may be many reasons for that, but what our work suggests is that one possible explanation is just that not all amyloid is the same. So just measuring total quantities of amyloid doesn't contain enough information. You actually have to distinguish what type of amyloid is a particular patient developing. So that, in order to do that, you need to develop, for example, imaging agents that can distinguish one fibril structure from another. Right now, there are PET imaging compounds that are used just to quantify total amyloid deposition. It's a little unclear what those are binding to and what specificity do they have, particularly in relation to the kind of structural variability that we've seen in our work. So characterizing the specificity of those imaging agents and probably developing better agents that have specific binding for the fibrils that actually do correlate with progression to Alzheimer's disease. That's the kind of impact that I think this overall line of research will have. I think potentially it could have a very large impact. It's going to involve, this is not just something that my lab can do. I mean, we're very good at determining molecular structures. We're not so good at discovering new imaging agents, but other labs specialize in that. So I'm hoping that the results that we've obtained to this point will encourage other labs to start working in this direction, start examining structural specificities of their measurements and specifically of imaging agents. I think is an important thing, and put effort into developing improved imaging compounds.